Exocrine uses ducts, endocrine does not. Good. Can you elaborate more on outside? Leave it just outside. You said something secreting outside. A little more.
Exocrine is on the outside, but on skin.
Surface of skin, surface of the surface of the body, and into hollow, into the lumen of hollow. Right. For example, gastric glands, digestive glands, are also salivary gland cells secrete into the ducts. Right. So body surface or into body. It's good. One more. What kind of secretions? Endocrine? Hormones, exocrine could be enzymes, could be. Oh, by the way, we'll go back to blood in a second. Say something.
What's the oil from exocrine?
Yeah, could be exocrine oil glands, sebaceous glands are exocrine glands, good. We mentioned that exocrine secretes body surface and lumen of organ. Where does endocrine secrete then?
To the blood.
Let's revise some of the theory we didn't talk about. Where can we find transitional in the urinary bladder, lining of the urinary bladder, and the ureters? What are they? What function? Yeah, to be able to expand and recoil, very good. How does that, because the name itself does not suggest it, but how does their structure allow us to do that? What about their structure? What is this transition? Are they one layer? Multiple, like they're just changing shapes, right? That's why it's transition. Squamous to columnar to cuboidal. Shapes, it's different depending on how we do it. Good. Where can we find stratified squamous epithelium?
Skin inside of your cheeks.
But skin, what layer of the skin?
Epidermis.
Epidermis of the skin, good. Is there something else? Vagina.
Something else.
Esophagus, and there's a cheek, so the oral cavity as well. Yeah, those all have stratified squamous epithelium. What's the functional significance of stratified squamous epithelium?
Protect against abrasions.
The underlying layers against abrasion. Let's look at simple column. Did we separate ciliated versus unciliated in this course? Where they're located and so on. We just talked about pseudostratified. Where can we find simple column then?
Kidney tubules.
Stomach to the rectum lining. What do we find in the kidney tubules? Cuboidal. So simple cuboidal we'll find in the kidney tubules. Where else do we find simple cuboidal?
Is the bronchi? Or the
Bronchi is simple column.
Alveoli?
Bronchi will be simple.
alveoli?
Glands. Alveoli would be what kind of tissue?
Simple squamous.
Simple squamous, right, that's the flat. That's what we use, that's the slide we use, that's pretty much. Those are ones for epithelium. Okay, so in terms of connective tissue, what's the division of connective tissue? What are some, first, before we go into the specific types, how would you group them? That was more in lecture rather than
Support.
They do support, yes, they can support it. Dense versus and loose, those are part of connective tissue proper, good. What else? What about the supportive connective tissue? That's our bone and cartilage, we'll talk about them. What other types of connective tissue? The liquid connective tissue, so we'll look at those two today. Let's summarize also our loose connective tissue. Yeah. Do we cover both dense and loose or just one? Both, so let's review. So loose connective tissue proper, we can divide into two, loose and dense connective tissue. What are the three types of loose connective tissue? Two of which we saw in lab, one we did. Adipose we saw in lab, areola connective tissue we looked at in lab, but if you're not looking at that, reticular connective tissue. So let's start with that one. Where can we find reticular connective tissue? The spleen, the lymph nodes, excellent. So they are basically good. They form what's called a stroma, kind of the what's the better way to describe, like the tissue chunky bit. But the tissue bits of the spleen, because we have spaces where we have blood, we have lymphoid tissue and so on. So these will form the kind of the building of it. So it helps to filter so less crabs. Where can we find areola connective tissue? Papillary layer of dermis, good. We can also find fascia and structures. What's the functional significance of this?
Stores.
Salt and water reservoir, good. Adipose connective tissue, where can we find them? Around organs, surrounding organs. What else? Hypodermis, which we'll talk about in chapter 6 today. Part of the hypodermis, which was looked at in the lab. Good. Functional significance. Here, let's name a few. Insulation, cushion, absorption, storing of triglycerides. Excellent. Good. That's our loose connective tissue. What are the three types of dense connective tissue? Dense regular, dense irregular, and dense elastic. Where can we find dense regular connective tissue? Tendons and ligaments. What's their functional significance? In one direction. That's going to be different from dense irregular because they can absorb straight in. Well, where can we find dense irregular connective tissue? Specifically, we can find them towards more the capsule of joints, but which part of the dermis? The reticular layer. Papillary layer of dermis is areola. Dense regular, it's going to be underneath reticular tissue. Good. Reticular layer of dermis. Next one, dense elastic connective tissue. Lung tissue, yeah. Arteries, veins, and trachea too. Yep, good. We looked at the arteries in our lab. What are their functions? Yeah, because elastic fibers are help to stretch and relax. What are the three components of all connective tissue? Fibers,
cells, and ground substances.
ground substance or extracellular matrix. Excellent. So fibers, cells, and ground substance, right? What are some fibers in connective tissue? So again, three different fibers. Collagen fibers, elastic, reticular. Which one is the thickest? Collagen. Which one can absorb the most amount of stress? Elastic collagen. Elastic is for elasticity. Thicker, elastic absorb the most tension. Where? So those fibers that we saw in our connective tissue that we saw in the artery to show dense elastic connective tissue, those black little fibers, those are the elastic fibers. What are some cells we can find in pretty much all of connective tissue.
Fat cells?
Fat cells, yeah, adipocytes generally spread throughout. Fibroblasts, that's an important one. What do they do?
Secrete.
The fat looks like fibroblasts. It's good. What else can we have? Immune cells, right? We can have white blood cells, and macrophages there. Good. Those will be our. By the way, is our connective tissue vascularized?
Not all,
Not all. Why do you say that?
some say vascular,
Like,
cartilage for your ear.
it's just the cartilage that's avascular, very poor vascularization, whereas all other connective tissue are very vascularized. What about epithelia? Are they vascularized? No, they are avascular, so they're depending on the connective tissue. They're close by underneath for their vascular support. Any questions? Okay, so we'll continue. So we talked about dense connective tissue there. We said the slide we finished on. So that one, so liquid connective tissue. So we'll go into more detail when we get to chapter cardiovascular. That's the first one, blood. But here's an introduction in terms of the connective tissue component. So blood tissue, we can separate them out into cells or the extracellular matrix, right? So the extracellular matrix is blood plasma, whatever those cells. The reason why I put them in quotation marks is one of them is not cells at all, okay? The other one is, it is a cell, but it cannot divide anymore. Only one really still has the proper functioning of the cell itself. Which one has proper functioning? Does anyone know?
White blood cells.
It's the white blood cells that do. Which one's not a cell at all? It's platelets. It's actually pinched off pieces. So in this slide, we see a couple of white blood cells, mostly red blood cells. And then a few, these little dots are actually platelets, which were pinched off cells. We'll talk about it when we get to blood chapter on blood. We'll talk about megakaryocytes. Okay. We also have, what are the functions of blood, by the way? Nutrients, waste, oxygen in terms of our, mostly our red blood cells, we talk about it, 98% of oxygen is transported within red blood cells. What else do we transport? CO2, mentioned waste, transport hormones, right? We talk about endocrine that secrete into blood, so they're transported by blood. We can also use it for, in terms of white blood cells, it's our immune response. We also talk about allergies. It's caused by triggering cells within the white blood cell, a type of white blood cell. And we also have response by another white blood cell, which is trying to dampen them down. So they can all have multiple different functions, right? As things like here in this image, the plasma would be in between the cells. What are some things that can be found in blood plasma? Because you might hear that we can transfuse blood plasma, we can donate blood plasma too. Who might be someone who might receive blood plasma?
Cancer patients, right, do they?
Cancer patients, not often, they could.
Surgeons.
Surgery,
Soldiers,
soldiers, why? What cases?
major trauma,
Why?
stem cells.
Because
Not necessarily stem cells.
because hypovolemic shock. Because if you lose the fluid, you can't adequately perfuse.
Yeah, and there's, yeah.
The blood clotting factors or like that?
Yeah, that's what helps with these. There are clotting factors in the blood which will help in terms of clotting. Yeah.
Hemophilia? Hemophilia?
Yeah, so they will receive that for the clotting factors because hemophilia, the different types, they are all lacking some one or two of the clotting factors. Something about blood volume, especially in a liver patient who may also transfuse blood plasma. The other ones are lymphatic tissues, right? So these are found in the lymph vessels, right? So here, I know we use it kind of loosely, lymph and lymphatic tissue kind of loosely, but if you're looking at lymphatic tissue and we separate out, the lymph is the extracellular matrix, the plasma equivalent of it. In terms of cells, that's just pretty much one cell that's carried by our lymph. It's just a lymphocyte. In terms of functions, they don't transport lymphocytes. Does everyone know what sort of nutrients are transported by our lymphatic tissue? Actually, two groups, two from different places.
Hormones?
Could be, but not usually not by lymph. But what nutrients are transported? So we will remove all proteins from interstitial tissue. So what is interstitial tissue? I mentioned in between cells, right? Within tissues or in between cells. So when from a blood vessel, some fluid leaks, some proteins may get through. If they do, the lymphatics are removed. Right? Lymphatics also transport fat from our intestines. They don't actually go into blood first. They go in the lymphatic system and then join the blood circulation. Left side. They'll join the blood circulation afterwards, whereas all other nutrients, whether it's on nucleic acids, the monomers of nucleic acids, carbohydrates, proteins, amino acids, the monosaccharides, nucleotides will go through the blood to the liver first. The fat actually. So we'll talk about that too. Okay, so those are the nutrients transported. Next, we're going to talk about cartilage. Okay, so cartilage is avascular. Okay, and they're sitting in a gelatin-like extracellular matrix. We do have fibers, some more visible in some cases, not so visible. Like the hyaline cartilage, it does have fibers. We do not see it under the microscope. Okay, it does stain a little different to be able to see it. So perichondrium is connective tissue that surrounds cartilage. Okay, it surrounds cartilage. It will provide the nutrition for cartilage, which is why if you rupture a cartilage, it takes a long time to heal because of this lack of blood supply. It is dependent on some tissue surrounding. Chondrocytes, these are the mature cartilage cells. So, who remembers what the hyaline cartilage looks like on the slides? Yeah, basically, if somebody took a crayon, purplish blue is what we saw. Crayon colored it with some spaces. There are some little circles there. Those circles had the cartilage cells, the chondrocytes in them. You might have seen a little dot. There might have been two sometimes in those what I call necruni, those little cavities, those little pockets in which they were sitting in this gelatinous extracellular matrix. Okay. So we have three types of cartilage. Let me actually, I know it's starting with hyaline, let's start with the other side. Because everywhere else will be hyaline. So, fibrocartilage and elastic cartilage are the other ones. There are three cartilages. So, fibro, elastic, and hyaline cartilage. I suggest learning these two first, but everywhere else is learning hyaline cartilage. I think we talked about elastic a little more in lab. Where can we find elastic cartilage? We didn't look at it, but I did mention. The ear pinna, the outer ear, is why it bounces back. Where else? The epiglottis as well. Hopefully you can tell me where's the epiglottis. Yeah, it's at the back of our throat. What's the function of the epiglottis? Get in the way of when we want to breath. tube down? Do we have to go? Might have talked about that. Yeah, so what does it, yeah, we don't want aspirate water. Yeah, so the epi means on top, epidermis, right, that's what epi means. We talked about epigastrium, right, epi means on top, glottis is basically the opening of the larynx, right, so it's sitting on top of the larynx, throat is the opening to the larynx, food goes down the pharynx into the esophagus rather than into our larynx, right, so prevents the air from entering. So this close the opening to the larynx called the glottis. Okay, so fibrocartilage we have in 3 places. Does anyone know of any? I know we mentioned one in the lab. Okay, so intervertebral discs. That's one. Where are intervertebral discs? Yeah, between the vertebral bones and stuff, right? So the vertebrae, the body of the vertebrae, which we'll learn in our next lab, we'll talk about structures of vertebrae. That's a big lab. Just look through those names before you come, because we're going to talk about a lot of them and find those landmarks. Some different, but you've heard of them, you know about them, some quite a lot. Maybe just Google the names of little things. So between the body of the vertebrae, we have the intervertebral discs. So if you heard of disc herniation or slip disc, it's basically this disc that we're all checking out. Good. The next is the pubic symphysis. Pubic bone is the bone in front of it's part of our pelvic bone is the one in front. So the 2 pelvic bones you meet in front. That is cartilage between them 2. That's fibrocartilage. That's the pubic symphysis. Third one is in our knees. What's that? The meniscus. Yeah, we have 2 menisci per knee. Meniscus singular, menisci plural. Those are made up of fibrocartilage. Okay. Fibrocartilage absorbs the most amount of shock. Elastic cartilage has a lot of elastic fibers, then it bounces back. Hyaline cartilage also absorbs shock. Okay, so the fibrocartilage absorbs the most amount of shock. So, where else can we have hyaline cartilage, or where else is there? The nose is hyaline cartilage. If you move your nose, you're not moving the cartilage. How much of our nose is bone? It's just the top is bone. Front all is all cartilage, and then the soft tissue. The only thing we can move is soft tissue. And we cannot move our hyaline cartilage. The ribs, they don't come all the way to the sternum. They stop kind of halfway. It's the cartilage, it's hyaline cartilage that connects the ribs to our sternum. Most of our ends of bones in joints, limb joints, are covered with hyaline cartilage. Where did we look for hyaline cartilage in the lab? What organ did we use? What other tissue did we use that same lab? Pseudostratified columnar. Where do we have pseudostratified columnar? More specifically, where did we look at?
Trachea.
The trachea is what we looked at. There, it's a C-shaped cartilage, that's what we saw. So our respiratory tract, so starting in our larynx, that got mostly hyaline cartilage. The only one is the epiglottis, which is elastic cartilage. There's a bunch of hyaline cartilage. If we want to put a tracheal tube from the outside, what's another name for it, or where do we go? cricoid, that's a cartilage hyaline, so it's between the thyroid cartilage and the cricoid cartilage. We call it cricotomy, right, or cricothyroid. So thyroid membrane. Those are all hyaline cartilage. Trachea has a nice C-shaped hyaline cartilage. Our bronchi do have, they get less and less as we go. Okay? So we have, that's what we talk about with respiratory tubes having hyaline cartilage. It helps to make sure that they remain. So precursor to bone, for example, our long bones. What's this bone here?
Humerus.
Humerus. So that's a long bone that developed first of all when we were very, very tiny as an embryo. It was hyaline cartilage first, which we converted to bone. We'll learn about this now, chapter 7. So you have hyaline cartilage first and then converted. Some of them remain, like the ends of long bones are still hyaline cartilage that remains. Okay. Fibrocartilage is 3 places: meniscus, pubic symphysis, and intervertebral discs. Elastic cartilage, outer portions of the ear, pinna, and epiglottis. Here, there's some elasticity bounces back. Here, absorbs the most amount of strain. Hyaline cartilage, we say it absorbs shock. This one absorbs the most amount of shock. But these are not very well designed. Into them quite often. But this does take up so much. I need 6 people on a daily basis. So in terms of bone, we in lab we looked at bone. What did the bone tissue look like? Tree trunk. So that's actually a type of bone. We have actually 2 types of histology of bone. It's compact and spongy. The one we looked at is compact bone. Okay? We'll talk a little bit more about this in the bone chapter. But we looked at the tree trunk. Okay. So these we'll talk a little bit more when we get to the chapter which is called an osteon. Those are units. We hope we saw many of these in that small piece. So here it's calcified extracellular matrix. Right? We'll talk much more about this in the chapter. So in terms of muscle, so muscle basically is to move things. Okay, we have 3 types of muscle tissue. We have cardiac muscle tissue, skeletal muscle tissue, and smooth muscle tissue. Let's start with the first 2. So skeletal, where can we find it? Attached to bones, right? Where can we find cardiac? Right. What about blood vessels? Is it still cardiac? It's actually the other one, the smooth one. So we always say, I always suggest to learn that in the walls of hollow bones, right? So our respiratory tract has them, our digestive tract has them, urogenital tract has them. What else did we see in lab that you may or may not recognize? I mentioned it's a smooth muscle. So smooth muscle we cannot consciously control. The lung tissue itself does not have. We have skeletal muscles around that help. Yes, skin. Which one? What structure is that? Raises the hair. Yeah, rectopili is a smooth muscle. Okay. So let's talk a little bit more about that. So skeletal muscle, if you haven't realized, it runs the entire length of that muscle. Each cell runs the entire length of the muscle. They're very skinny, they're almost thread-like, so hence a muscle cell is also called a muscle fiber. Okay, and they run parallel to each other. Another thing is these are multinucleated. What does that mean?
Multiple nuclei.
Multiple nuclei. They have striations. Striations, you can think of it like the zebra stripes. They are banded. They have dark bands followed by light bands. Okay? And I'll just talk about another one that is so. Once we've reached adult size, they are incapable of dividing. What does that mean for us?
You can't make new ones?
Yeah, you can make new ones. But what happens when I go to the gym, if I want to bulk up, I keep working out, and the mass increases? How did we achieve that? The number of cells do not change, but the size of the cell changes. That's what muscles, as an organ increase in size. So we do not divide muscle cells. Skeletal muscle cells. Same thing with cardiac muscle cells. While smooth muscle cells we can divide. But these two we do not. What else do I want to talk about? And are they under voluntary or involuntary control? Voluntary. So voluntary control is conscious control. Don't say we can't control them, because yes, our heart muscle is controlled by us. It's not voluntarily controlled by the brain and control. Right? So this is the only one we can voluntarily control, consciously control it, while the other two are involuntary. Okay? So here, this is a drawing of a muscle. So it's 3 muscle cells. These are very skinny. They could even run in not just this wall. We went to scale it up. It will run the entire length of these buildings. Quite big muscles, long muscle cells, not big, very thin, long cells. They run the entire length, even up to half a meter long. Bigger muscles, especially taller people, very long muscle cells. Here, it's showing them again. These are really 2. That's 1 full. Again, going in that direction. Horizontal direction. You can hopefully appreciate, hopefully you can see it from there, especially on this side. There's some banding. It looks like little lines, red and like pink in between. That's the striations. Okay. In this cell, for example, we already see 2 nuclei. This is a very long cell, so they are multinucleated as well. Okay. So cardiac muscle only found in the heart wall. These ones are striated. They are involuntary. Mostly 1 nucleus per cell. Some cells do have 2. Okay? And this is the only muscle cell which is branched. Okay? So here it's showing a branched cell. It's kind of spreading. That's only cardiac muscle cells are branched. Okay? I'll talk about intercalated discs in a second. But again, mostly contain singles. So 2, some of them do contain 2. They're branched, they're involuntary, they are striated. They're short cells. Okay, they're not very long cells, so they have to be connected to other cells. We don't want to have to stimulate every single cell, and we want the whole chamber to contract as a unit. Okay, so they have junctions between them are called intercalated discs. These will allow, what is impulse, nerve impulse in a nervous system, basically.
Is it when a synapses happens? Is that what
So
it
at
is?
the synapse, it can trigger it, sure.
Signal.
It's a signal, it's an electrical signal, it's an exchange of ions. So if we have some extra ions here, we've stimulated this has, we'll talk about it's called action potential. If we have stimulated an action potential here, through this intercalated disc, those ions from here can go to this cell and cause an action potential there. So this way we don't have to stimulate each one. Okay? That will be the significance of intercalated discs. These are the only ones that have intercalated discs. Smooth muscle do have those junctions between them which allow ions to pass through, but they're not intercalated. They're not these nice, very thick connections between 2 adjacent adjacent cells. We'll talk about this in a So you may have heard of permanent pacemaker, right? Who gets a permanent pacemaker? What happened?
I say no doubt.
Yeah, or does not communicate. So we have parts in our heart which are called pacemaker cells. This function is not to contract, but to actually start the impulse themselves without our nervous system coming in there and stimulating. So we'll talk about different parts. We have a pacemaker when either the parts are not connected or not communicating well, and then hence they're firing at random points, or one is not working at all in the pacemaker. We take over because we don't have to send an impulse each time for the heart to beat. So we're beating it about 60 times a minute, which means we have to send the electrical activity to the heart 60 times a minute, right? Increase it during stress. So that's when our nervous system plays a role. Here, it can trigger its own acceleration, its own nerve impulse. So we'll talk a little bit more about that as well. So here, to summarize, they're short. They are branched. That's my branch. They are branched. They have generally one nucleus per cell, sometimes two. They are striated. They are involuntary. Okay. What is involuntary?
Can't consciously control it.
Not consciously controlled. They're still controlled by our brain. These ones, when they're not nice state, when it's actually this is from obviously a dead person or dead tissue here, when you don't necessarily see the striations very well. They actually are very good at just collapsing on itself. You don't see the striation. There's one spot here, you can see some striations. Not as good as another that slide to show that. This is histology, this is a drawing. Here they have shown the striations better. You can see some back here, like that, like the dark bands. It's because they collapse on itself. There. Smooth muscles, if I ask where the location is, say walls of hollow organs, right? So, digestive tract, respiratory tract, urogenital tract, all we do have smooth muscle in the walls. We mentioned the erector pili. What about in our eyes? Are there smooth muscle in our eyes? The iris, the diagram here, the iris of our eye is smooth muscle as well, right? So we do have other places, but this has a big one phrase answer, walls of hollow organs. They are spindle shaped, so they are narrow at the ends, wide in the middle. Okay. These ones only have one nucleus per cell. Okay? One nucleus per cell. Another thing is they are involuntary. They do not have striations. Okay? Involuntary, they do not have striations. These are the only cells, even ones mature, can divide. The other two do not. Skeletal and cardiac do not divide. Okay, so it's only by question read which muscle cell has only one nucleus per cell, then you're looking for smooth muscle, which one always has multinucleated cells, skeletal, because cardiac mostly one, but they have two nuclei per cell. Which ones have striation? Cardiac and skeletal. Which ones are involuntary control? Cardiac is smooth all the time. Is there any involuntary control of our skeletal muscles? Like shaking? When is there involuntary control? Yes, yeah, sure. What kind of response? A reflex. So it's a reflex response, right? So if you there are other responses, other reflexes too, right? You touch a very hot object, your brain, it's really, your spinal cord is already stimulated and to come out even before your brain is realized. So reflexes. Is diaphragm a skeletal muscle? Can you breathe faster? Can you breathe slower? Yeah, we can. But our brain try to stop and hold your breath for a little while, we can, but can we hold it for a long time? No, then our reflex will kick in, right? But diaphragm is actually a skeletal muscle. Singing, you're using the diaphragm under your own voluntary control. So skeletal muscles are voluntary control, but also involuntary activity. Any questions? Okay, so the last of the tissues is our nervous tissue. Again, here is just an introduction. We're going to go into more detail in that chapter. Please realize there are two separate, very distinguished cell types. One is the one we always think about function in our nervous system, the one that sends those impulses, right, or carries those impulses. That is the neuron. Okay, but we have cells that are around neurons, connecting neurons, surrounding them, that help the neurons. These are grouped together as neuroglia or neuroglial cells. One of this group divides, the other does not. Do you know which one does not divide once we reach adult size? Neurons. So we kill the neurons, that's the end of it. Does not replace it. But neuroglial cells cannot. Which means, which ones will give rise to cancers? Neuroglial cells, neurons do not, right? So neuroglial cells that give rise to cancer because they're the ones that divide, neurons do not divide. Have we heard of Schwann cell? That's the one that people have heard of neuroglial cells the most often, but in terms of cancer, they've possibly heard of others. The Schwann cell produces myelin. Okay? What is myelin? Myelin sheath. It insulates and surrounds part of the neuron. Yeah.
Does it increase the speed of the sheet drug, right?
Yeah. It insulates and increases the speed of the impulses. Okay. So they support these, we'll talk about some of them. They support, they help maintain the neuron. So yes, we cannot replace the whole neuron, but if a part of the neuron, depending on where, sometimes we cannot, if a certain part of the neuron got cut off, there is potential to repair, okay, but not the whole neuron cannot be replaced. We'll go into more details in chapter. Here, let's describe a few things, right? So neuron, we can first learn as it's got three parts. First, it's got a cell body, okay? Cell body contains the nucleus and most of the organs, okay? And then we have little what I call processes that are attached to the cell body. And these processes are named by what direction the impulse goes. So dendrites, branching on dendrites, these will receive it. So impulse will come from by the dendrite and go to the cell body. Okay. This one is a nice one that you can see short dendrites, but that's not a requirement. Okay. The requirement or definition is dendrites take impulses to the cell body. Okay. While the axon takes the impulse away from the cell body. Okay. So as an introduction, we'll learn there are three parts. Don't say that nowhere else do we not have organelles. We do have organelles, especially in long axons. We can have mitochondria and other sorts, mostly mitochondria, but we have granules and so on. But most of the organelles are in the nucleus, most of the endoplasmic reticulology. What was the function of? People were still confused. I finished grading, but I started grading the quiz. What's the function of rough EPAR? What are functions? Number 2. Produces proteins and phospholipids. What are functions of smooth ER? Triglycerides, steroid hormones, detoxifies drugs, which is why our liver has a lot of smooth ER, in the cells have a lot of smooth ER. They are found at the cell. Okay, it's larger than the rest. You can actually see the largest one is the pyramidal cell, put it under a microscope, under which power. We don't have to get to high power. The low power, this whole cell body will cover the field of view. Okay, that's one of our bigger ones. The oocyte is another big cell body. Next, we're going to talk about what are called membranes. So here, membranes, we have epithelium and the connective tissue forming a unit. Most books at least have 3. Our book describes a 4th. I'll tell you why and why some books do not have that. So mucous membranes, main function or one of the functional significance is produce mucus, right? So it's going to produce that mucus. So respiratory tract has mucus. Urogenital tract has mucus membrane. Digestive tract has mucus. While serous is a slippery fluid. That's we did talk about the cavities. What's the cavity holding just the lungs? Pleural cavity. Pleura is a serous membrane. Okay. What's the one that surrounds just our heart? Pericardium. That's a serous membrane. What's the one that surrounds organs in our abdominal cavity? Peritoneum. Those are all serous membranes. We have 2 layers of serous membrane. Inner layer is called the visceral and the parietal. Good. Cutaneous. We didn't mention that. What do we think that might be? The skin. Yeah, that's our cutaneous membrane. So what are the 2 layers of the cutaneous membrane? Again, we need what? We need an epithelial layer and a connective tissue layer. What's the epithelial layer of our skin? Epidermis, very good. And the dermis becomes the connective tissue. Is hypodermis part of our skin? No, technically it's not actually part of our skin, it's just underneath. Hypodermis below the skin. Okay. Synovial membrane. Does anyone know where that can be found? Yeah, and we've been joined. We're talking about synovial cavities. It forms this capsule around with it in a joint. This is part of it. This is the one that some books do not describe because it's not complete epithelium. It's a lot of connective tissue with patches of it, which is why some books do not describe it. Here it is. Here we do have it, so it's best. Okay. So mucous membranes, right, does produce mucus. Do we remember what those cells were called that produces the mucus? Goblet cells. Those are unicellular glands. They produce mucus. What's the function of cilia?
Move mucus.
Move mucus, move substances across them. The cell surface mucus. In the fallopian tubes we have cilia too. What are they moving there?
The egg.
The ovum or the oocyte. Good. Okay. What else? Good. So epithelium on top, and then the connective tissue underneath. Lamina propria it's called. It's mostly areolar connective tissue. Okay. So these 2 together will be the mucous membrane. By the way, where do we have simple column epithelium again?
The lining of your stomach to your rectum.
Lining of stomach to rectum. Where else? Fallopian tubes. One more. So I want to distinguish between pseudostratified. Where do we find pseudostratified column epithelium? The trachea and large bronchi. Well, the small bronchi will also have column epithelium. And serous membrane, this is one which again we have epithelium and connective tissue, but here main focus that slippery fluid between them produced by the branch. So it is, in terms of cells, the predominant cell in this loose connective tissue, you might have heard the cancer associated with that. It's a cancer of the lungs, the lining of the pleura, especially in asbestos.
Mesothelioma.
Mesothelial cells is part of this epithelial cells, and that's the mesothelial cells, which will produce the serous. So that's where mesothelioma name comes from, because it's cancer. Or the serous lining is really of the pleura around lungs. So two layers, parietal versus visceral. Visceral layers right on the organ, and in between there's a little space where we have the serous fluid, and then it's the parietal layer. Okay. Inner surface, we're talking about the surface of the body, but not necessarily the surface of that organ, right? This one covers the surface. Cutaneous membrane, that's our stratified squamous epithelium in our epidermis. And then, what's the top layer of the top layer of the dermis called?
Papillary?
Papillary layer of dermis. Which loose connective tissue is in there?
Areolar connective tissue?
Areolar connective tissue here, and then dense, irregular connective tissue in our reticular layer of dermis. Last one's our synovial membrane. Here you can kind of see epithelia, these red cells. It's not continuous, there's some spaces where there aren't any epithelial cells. This is probably the knee that they're trying to show. Here is forming the cavity, so it's on the outside, right? You have bone here, bone there, the outer surface making a cavity within that joint. That's the synovial membrane. They produce a synovial fluid, which is also a slippery fluid. It's like our brake fluid in our cars, right? It's not the brake fluid that helps the brakes to work, right? The brake pads that stop it. But we need those brake fluids to keep it smooth. Same here, movement. It's not the synovial fluid themselves that just the movement, but it helps smooth those movements out. Okay. Any questions? The last thing we're going to talk about is regeneration. So realize some have very good regeneration, some have poor, some have very much no regeneration. So let's look at the good ones, right? Epithelial cells have good regeneration capacity. They need to have good regeneration capacity. Why? They're exposed to the elements, whether it's outside of our body or within, say, our digestive tract, or it's food's passing through, water is passing through, they're exposed to that. Right? So they need to have a good regeneration capacity. Okay? Bone has a very good regeneration capacity. Right? Break our bone, yes, it takes a little while to heal, but when it heals, actually we put down a thicker bone there. Okay? So very good regeneration capacity. Areolar connective tissue, dense, very important, irregular connective tissue has good regeneration capacity. Or dense, regular, if you rupture the tendon, you rupture ligaments, you know how it took a long time to heal. Right? Some of them heal by themselves, but a lot of times we needed some sort of surgery to keep the ends together. So they are more regular. Okay? Blood forming tissue or blood cells will be good. Smooth is good. Cardiac, skeletal, here they put it under poor regeneration because not because the cells themselves can divide, it's because the cells can heal, they can increase in size. That's why it's poor regeneration. The number of cells do not change or do not increase, or decrease over time. And cardiac muscle and nervous, and more specifically, what type of nervous tissue does not divide? Neurons, neuroglial cells do have good. They're kind of in this class of good class, so it's not here when they talk about nervous tissue, they're really talking about the neurons. Question?